Now, we have to know what kind of compounds are stored inside the human body in order to be used as a source of energy later on. So we mentioned that we have glucose, and if we have an excess of glucose, it's converted into glycogen, where it's stored in the liver, in the muscles, and so on, and it can be broken down later and be used again as an energy source to go through the steps that we mentioned before. We also need to know that we have other structures. For example, we have fatty acids. They are stored in the adipose tissue. So they are important in the formation of adipose tissue. We have cholesterol, which is important for the formation or the synthesis of structures in the cell membranes. We also have polysaccharides. Polysaccharides are essential for the structures in the intracellular matrices, and so on. So the structures, the inputs that we mentioned in the beginning of the video, can be stored in these formations, and later on they can be broken down to the initial or the basic structures, like glucose. We mentioned it changes into pyruvate, the proteins into amino acids and fatty acids. All of them will be converted into acetyl-CoA in order to produce the energy once more. But the thing is, we have some kind of an order for the body to use these inputs. So glucose is the first one to be used in this metabolism, followed by the fatty acids. And proteins are not used normally in the normal diet, so they can be used, but in cases of prolonged fast. So proteins are the hardest to be used in this process because they are important for other processes. They're important for other structures like contractile elements in muscles, some elements in the intracellular matrices, and so on. So this is the order through which the structures will be used in order to obtain energy. Finally, I'd like to talk about the regulation of this energy metabolism cycle. We have to know that throughout the cycle, we have regulating enzymes, which are called rate determining enzymes. So for each step we have this enzyme or couple enzymes which are crucial. So their increase or decrease would determine the proceeding of the step or stopping the process. So these enzymes themselves are regulated by two methods. We have something called allosteric regulation. So we have these compounds which are, if found, they would affect the enzyme in a positive or a negative way. And we have insulin and glucagon. Insulin and glucagon can induce the phosphorylation, dephosphorylation of these enzymes. So this is direct interaction with the structure of the enzyme itself. So it phosphorylates or dephosphorylates the enzyme, and depending on each enzyme, the phosphorylation could actually activate or deactivate the enzyme, and we will talk about this later. So we have these two kinds of regulation for the rate determining enzymes. And we have this other kind of regulation which is related to gene transcription. So gene transcription, which is also affected by insulin and glucagon, would affect the rate determining enzymes in an indirect manner to activate or deactivate the step. So briefly, this is how the regulation of these energy metabolism steps go. And that was a brief idea of what we're gonna talk about in the next videos. And in the next time, we're gonna talk about types of nutrition and how do they affect this energy metabolism cycle. So I hope the video for today was easy and clear, and until the next time, I thank you for watching and see you.